Hello, Georgia. Can you unmute and say hello to everyone?
Hello. Hi, Jack. Hi, everybody.
So thank you very much for joining us, Georgia. As I said, you came to the Wellcome Genome Campus in 2020, and your work then was quite different to what it is now. But tell us about what it is that you're doing now within the Genomics Surveillance Unit.
Yeah, so at the moment, I work in the Genomic Surveillance Unit and specifically in the parasite team. So I look at how parasites change over space and time and how this helps us tackle the disease malaria, which we'll be focusing on today.
Fantastic. Thank you very much, Georgia. And what would a normal day look like for you working in the Genomics Surveillance Unit?
That is the worst question to ask a data-y person because every day is so different, but that's a very cliche, rubbish answer. So days are very mixed, and I mean that in terms of the work and in terms of how we do the work. So I always used to think that a computer science person was very isolated, stuck behind a computer all day. But a lot of my job is very talkative, very collaborative. I meet with my teammates. I meet with partners overseas via Zoom calls to kind of discuss ideas and analysis that we're working on. And I do spend time at my computer coding away on DNA data.
Lots of people, me included, hear coding and can sometimes feel a little scared of it or think that it's a difficult thing to get into. But I know for you personally, you've had an unusual journey into this job that you've got to. When did coding become something that you did as part of your day-to-day job, and how did you learn to do it?
Yeah, so I studied an undergraduate degree in genetics, which was completely wet lab. It wasn't computational based. Coding wasn't part of my curriculum. But it was in the summer of my second year that I decided to have a go at teaching myself how to code in my free time. Sounds very nerdy. But I started teaching myself how to code in my free time. And then I realized this fantastic world exists where we combine biology with informatics, and we have this whole field of bioinformatics where you can then apply coding skills to biological information. So that's how I transitioned into using coding in my everyday life now as a data scientist, because I taught myself those basic skills and then I was able to then apply them learning on the job through my current career.
Fantastic. We're going to hear about your time here on campus through two of the main focuses of your time here. Give us a snapshot of your journey to campus, because you've worked in some really interesting places before coming to here. Yeah, I'd like to give everyone a sense of that journey.
Yeah, so I mean, interesting places. During my undergraduate degree, I managed a cocktail bar. That was my first job. So yeah, I was managing a full-time job whilst doing my undergrad, which meant that, yeah, a lot of my time wasn't really focused on thinking I was a scientist because I spent so much of my time working on something else. But that doesn't mean that in the future I wasn't going to become that scientist that I am today. So that was a really important thing that I learned from that period of my time. Even if you don't do something full time, doesn't mean that you won't one day. So I was working in a cocktail bar during my degree. And then after I finished my degree, I was then able to have an internship in bioinformatics. So these were normally saved for people during their degree, but I wanted one and they had a space and I applied for funding. So I did an internship funded by the Genetics Society, and it was hosted at the Earlham Institute on the Norwich Research Campus, which is a wonderful campus with many great institutes on there doing some wonderful life science research. So I spent my summer there, getting better at bioinformatics, actually applying my skills in the real world, surrounded by people working in a bioinformatics lab. So I spent the summer there and then ended up on campus in the October of that year.
Fantastic. Well, thank you very much, Georgia. Name check a couple of brilliant organizations there. So anyone on the call who's interested in genomics, of course, explore Wellcome Connecting Science. of course, explore our website, Your Genome, and I'll be putting a link up to that at the end. But also do have a look on the Genetic Society website. They've got some fantastic resources and programs going on there. Do explore Earlham Institute, particularly if you're in and around Norwich. Some fantastic research and projects going on there too. So yes, I'm glad we got to shout out to those two. But at this point, I'm going to ask you to get your slides ready, Georgia. And for everyone who is on the call, we're going to be sitting with Georgia through this brilliant account of first COVID surveillance and the development of skills there, and then taking those skills that Georgia developed in that work into malaria surveillance and the different challenges too. So Georgia, if you're ready, I'll hand over to you.
Yeah, sounds wonderful. So can you see my slides? I'm going to presume yes, unless nothing gets shouted out. So thank you everybody for joining me for today's webinar. So as Jack has lovely introduced, we are going to be looking at mapping malaria with this thing called genomic surveillance. So my name is Georgia Witten, and I'm a senior data scientist here on campus, working in the genomic surveillance unit, which we'll come to understand what that means through the course of this presentation. So what I'll be talking you through today, first of all, I'm going to outline the disease malaria, so we all understand what it is and the burden that it places on our world. I'm then going to highlight how genomic surveillance was used in the COVID-19 pandemic, because that's something that we can all be familiar with as it was on our very doorstep. I'm then going to talk about the specific challenges that we have when trying to use genomic surveillance in malaria. And then I'll be discussing a use case of genomic surveillance for malaria. So, the burden of malaria. Malaria it's a life-threatening disease, which is something really key to keep in mind. And malaria is a complex disease because there are kind of three key components that we have to think about as scientists. So we have a mosquito, which is a vector, which carries a parasite called Plasmodium. And then this mosquito will bite a human and infect them with the parasite. And then it's that parasite that makes you sick during the malaria disease. There are 5 different species of this parasite, and the main one is called falciparum. This is the most deadly, and this is the type of malaria parasite that we focus on the most during our research and genomic surveillance activities. What's really key to note is that malaria is preventable and it's curable. Though sadly, nearly half of the world's population live in areas that are at risk of malaria transmission. And this includes 87 countries and territories. And it disproportionately affects Africa and young children, which again, a really key thing to remember when we're moving forward, thinking about why this is a very important disease to be tackling with the computational tools that we have. It's also causing a lot of money in GDP to be lost from the countries that are facing this disease. So take homes, we have a mosquito carrying a parasite that bites a human, then they get infected, and it's the parasite that makes them sick with the disease we call malaria. This is what the clinical presentation would look like for somebody presenting with malaria. So we can see themes here like fever, chills, headaches, muscle and joint pain, rapid heart rate, rapid breathing. And you can imagine it can be quite difficult to specifically diagnose somebody presenting with malaria because it's difficult to disentangle these symptoms from other diseases in non-endemic settings. You can then have more severe malaria when the disease progresses, resulting in things like confusion, neurological focal signs, and severe anemia and respiratory difficulties. So here I'm showing you a graph of the age mortality. So we're looking at the amount of people that die from malaria every year, and we're looking at the age distribution. So let me just orient you to this graph. So this axis here on the horizontal line is progressing in time. So we've got 1990 here, going all the way up to 2000 and 2019. And then the vertical axis here is the number of deaths by age for that year. And then the color of this graph represents the age group. So we can see here, the colors are split by the age group. And sadly, we see under 5s takes up the majority of deaths from malaria every single year. So a very, very hard-hitting statistic to be aware of when we're thinking about how important it is that we use our resources to tackle this disease. And there are things we're doing to tackle malaria. We have treatments and we do have interventions. So I mentioned we have the mosquito and we have the plasmodium, the vector and the parasite, and we have different preventions and different treatments that we can use when trying to fight off the disease. So, for example, for the mosquito, we can treat bed nets with insecticides, which stops the mosquitoes coming into somebody during the night and biting them. We can also use indoor residual spraying, so spraying the air to deter the mosquitoes. Then for the parasite, the plasmodium, we also have some preventative treatments. So we can administer drugs to people who are highly likely to get very sick from malaria. So people who are pregnant. And then in terms of treatment, we have a treatment for treating the parasite, because as I mentioned, and key to remember, it's the parasite that makes you sick. So when we're giving treatment, the treatment is targeted at the parasite. So we have lots of drugs to treat malaria, and we call these anti-malarials. There are a few of these that are on the market, such as chloroquine, artemisinin, and other names that we don't need to go into for the contents of this talk. So, using genomic surveillance to track the spread of COVID-19 pandemic. So I've given you the introduction to malaria. So we've now got an idea of what the disease looks like and the treatments and interventions that we try to apply in the field. But this talks about genomic surveillance. So I'm going to introduce you to the term genomic surveillance and illustrate this through the COVID-19 pandemic. So what do we even mean by this term genomic surveillance? So when we say genomic, we're talking about the DNA material of an organism. So every organism has its own DNA material. And when we say genomic, we're talking about that. Then the word surveillance. So surveillance means watching for changes through time and through space. So genomic surveillance is watching for changes in the DNA material of an organism through time and through space. There are lots of really helpful things that we can understand from genomic surveillance. We can track changes, we can do outbreak detection, we can cluster organisms into similar groups, we can identify new variants, we can monitor changes after we've released things like new vaccines. We can do historic analyses, so we can go back and look at our historic surveillance data and see if there were any signals that we missed. And then we can also understand things like population structure. So take home from this, there are many helpful things in terms of epidemiology that we can learn from genomic surveillance. And now we're going to see genomic surveillance in action. So I'd like you to take your mind back to October 2020. And apologies for people not living in the UK. This will be a little bit more familiar with people living in the UK, but it's still a really good example to watch how genomic surveillance was working in a country. So we're back in 2020 and genomic surveillance was being used to track the different variants of COVID that were spreading throughout the UK. So we could use this information from the variants, from those samples, and then see where the variants were within the country. So the team at Sanger developed a computational tool in order to visualize these variants spreading across the UK. Very, very interesting to watch. So we're going to watch a clip from this first variant. And I'm going to play it twice because it's very quick. So you need to really keep your eyes peeled at the image of England. So we want to see where the variant first appeared. So I'll play it once and then I'll play it again. So currently everything's in this yellow color, and we're going to see the color change over time. So at the top here is progressing through time, and then each kind of division is colored by the amount of variant in that district. So I'm going to play this once for you. So as we're going through time at the top, you can see that suddenly, as we progress through time, most of England is then covered in purple, denoting that this variant has spread all across England. So I'm going to play it one more time. And really keep your eyes peeled. Where did this variant first appear and then spread through England? You should have the poll on your screens now. So how eagle-eyed were you? Where did this variant first appear in around October 2020 in the UK?
We look to have a good number of people completing that poll at the moment. I'll just give people a few seconds more if they want to complete it. For some people, if you're watching on a phone, you might not be able to access the polls. If you still want to take part, you can type things into the chat if you want to put it there. So if you want to put your answer in. But I'm going to share the results now. It looks like we've got a pretty clear winner here, Georgia.
Oh, so fantastic.
Yeah, one person thinking Hartlepool and one person thinking Devon, two people thinking Herefordshire, but everyone else thinking Kent in the southeast of England.
Ah, looks like people are paying attention, or maybe they remember, but yes. So that was the correct answer. The answer was Kent. So for the few people that didn't get it, I'm just going to quickly play it. You see it pop up in this corner first, and then it spreads throughout the UK. So by having the sequences of these variants, we were able to see where they appeared in the UK first. So very, very great stuff that genomic surveillance is able to show us. So, and I have a question for you. So if you guys post in the question and answer function on Zoom, so this variant appeared in Kent and it was called the Kent variant. And it cancelled Christmas, if we can remember, for people living in the UK. But can anybody remember the scientific name for this variant that was then spread through the UK during Christmas time?
We have had one answer come in. I'm just going to give a second in case anyone else too. This is in the chat function. Yeah, we've had another answer in now, different answers actually.
You can see the theme of the answers, the Greek alphabet. We're on to something here.
Well, just to clarify, Georgia, our participants won't be able to see these answers coming in. So I'm going to read a couple of them out. We've had a few people say Omicron. We've had a few people say Alpha. We've got Delta, and we've got someone who said B117. So which one is right in this case, Georgia?
So these are all names of variants, so very good. But the Kent variant specifically was called the Alpha variant. So yeah, this one was the Alpha variant, and then Omicron came later. So yeah, known as the Kent variant, but then scientifically known as the Alpha variant. So this really was a genomic surveillance success story. I do want to highlight that genomic surveillance was around before the COVID pandemic. We were working on it for malaria prior to the pandemic, but the pandemic really highlights how much action we can have when this successfully gets executed, with lots of pressures on people to come together at once and get something done. So during the peak of the pandemic, we were sequencing thousands and thousands of these viral COVID samples. And at the peak of the pandemic, this was up to 66,000 a week. So 66,000 a week samples were arriving to our genome campus, and we were sequencing their genome. So an incredible amount of surveillance activity going on. And what we've just witnessed and spoken about is the sequence information. So understanding these different variants was then be able to be used by policymakers to make decisions about public health. So imposing lockdowns, conducting arrival testing, doing flight bans, we could make these decisions because of genomic information. So before we move on to malaria, we're going to have another quick poll because they're so fun. So I want you to picture this moment. You are sitting in a lovely cottage. Here you are in the Lake District in a hot tub, looking out at your lovely, beautiful view. And then suddenly you feel a bit of a tingle in your throat. You start to have a bit of a cough and you think, do you know what? This probably could be COVID. I should go and take a test. So you get out of your hot tub and you head to your local test center in the Lake District, and you take a swab in your car, give it to the man in a fold-away bag, and then that test gets sent off for DNA sequencing. And what would happen to that swab that you gave to your local test center in the Lake District? It would travel all the way from your hot tub in the Lake District, round the country, and then to our genome sequencing center on the Genome campus in Cambridge, and then have to be genome sequenced and then uploaded to a collection of those sequences. So the next poll that's going to pop up on your screens is, how long do you think that it would take from taking that swab up in the Lake District, your sample then traveling all the way over to Cambridge, over to the Genome campus, us then conducting DNA sequencing on that sample, and then doing the analysis and uploading it to a sequence collection that we have here? So we've got 4 options for you. It's either, is it 3 hours, 2 days, 1 week, or a month?
We've got a good variation on the answers coming in here, Georgia. I can see we've had a really good number of people engaged with this poll, actually. I'm just going to give it a couple seconds longer if there's anyone who's still deciding. But I am going to end the poll there and share the results. So quite a spread, you should be able to see. So we've got 4 people said 3 hours, 29. So the majority actually said 2 days. We had 10 people say that it would take a week, and 2 people said it would take a month. So which one of those was correct, Georgia?
So the one that was correct out of those was 1 week. So if you kind of break it down and think about where your swab would have had to have gone. So you'd have to take that swab, and then it would then be sent to a first laboratory where they'd give you that text that says you're COVID positive or you're COVID negative, because you need to know that information straight away. Then these are then sent all the way over to Sanger, and then we have to conduct DNA sequencing. So if we break this down, the first step, so having your test taken and then getting a positive, negative result takes about 1 to 2 days. So the people who said 1 to 2 days, that does work if we're thinking about just having a positive or negative test. But to do the actual DNA sequencing and find out which variant of COVID that you had, that would take much longer because we have to conduct a very complicated lab process on that sample to get that sequence. So doing this in 7 days is a fantastic turnaround time when we're thinking about actual real-time genomic surveillance. And that's what you need if you want to make decisions about public health. You have to have information that is accurate and relevant. So it has to be timely. So we'll move on from this poll. I'm going to ask you to put some answers in the chat function to a second question. So we've seen how COVID pandemics worked in terms of genomic surveillance, but why might it be harder to conduct genomic surveillance on malaria? So thinking about the malaria disease, we've touched on a couple things at the beginning of this talk already. So why might it be harder to do genomic surveillance on malaria compared to COVID? So yeah, feel free to post in the chat.
Yeah, we've had some good things coming in here. Thank you, Georgia. I'm going to read these out as they come in. Yeah, great. Some really good thoughts already. So we have logistics of collecting samples, multiple species of Plasmodium. Thank you. Because the countries affected are mostly in low and middle income countries. That's great. It's carried by mosquitoes. Yeah. So I'll be interested to hear that challenge. Diagnosis differences, remote geographical areas, lack of funds, lack of trained staff. Too many people have it and take it for granted. There's loads of other really brilliant options here, and I'll see how many of these come out in the things that George is going to say over the next few minutes.
Yeah, a lot of fantastic comments there. You guys could have done this presentation for me, judging by what you've said in the chat box, which is fantastic. So let's carry on now, and we're going to spend the final part of this talk today talking about genomic surveillance for malaria. So when we're thinking about genomic surveillance for malaria, and when I say we, I'm referring to the community, we need to think about, can we sequence these thousands of samples to give us the bigger picture? Can we identify these variants that are concerning? And then can we make ways of giving this information that we have to policymakers to enable them to make decisions that are based, or well, not based, make decisions that are contributed to by the information from genomic surveillance. So we want to basically inform decision-making with genomic data. So we saw from the COVID pandemic story that there are things that we need for successful genomic surveillance. So we need lots of data, and we need it to be relevant. We need to deliver the data back in real time. So we talked about that 7-day turnaround in terms of COVID. So we need to be having real-time turnaround so that the information is relevant, and then you can create information that is timely, which is very important. And then we need good geographic coverage. It's only by having a good spread of samples that you're able to really identify those important epidemiological things that you need when thinking about genomic surveillance. So in terms of malaria, there are several things that can make this difficult for us. So the parasite genome size. So the COVID genome is pretty small when it comes to genomes. The parasite genome is much, much larger. It's around 738 times larger than the COVID genome. So this means that there is, first of all, much more data, which requires storage somewhere. So you need to have access to larger compute resources to store this bigger data. It's also much more complex to analyze because there's more in there that we need to be able to understand how to work with. So you need to have the analysis skills and techniques to enable you to draw conclusions from this larger genomic data. And it means that you often need kind of high performance computing systems in order to accurately analyze lots of these genomes because they're so big. And it also means that there's more chance for things to go wrong. If you've got more genome size, if you've got more genome length, then there's more space for variants to pop up. There's more places things can go wrong. So finding the relevant data within that genome is very important. Then when we're thinking about delivering the information in real time, so the issue we have in malaria, or historically we have, is the location of sequencing. So historically in malaria, people would ship back their samples overseas into the UK, and then we could sequence their malaria parasite genomes for them and then send them back that information. But if we're thinking about decreasing turnaround times, you know, having to ship a sample out of country to another country to then do that sequencing is not going to deliver you real-time information. And then we have things like sample spread. So we need to make sure that we're having that good geographic coverage, but malaria samples can be located in difficult to access regions. We can have false negative testing. So sometimes somebody might have a malaria test and it says that they're negative, but they're actually positive. So we can then miss these cases. So there are things in malaria, and this is just a few of them, that make genomic surveillance difficult. But there are things that we can do to make this successful. There are ways that we can adapt what we're doing and make sure that we can do genomic surveillance on this very complicated disease. So in terms of that parasite genome size, we don't have to sequence all of it. We could just sequence the areas in that genome that we're interested in for genomic surveillance, the areas that are relevant to the certain metrics that we're looking at. So we don't need all of the information. We can just sequence parts of it. And this means that it can be quicker, it can be easier, the data is smaller, takes up less space, it's easier to analyze and therefore easier to interpret, which is important for having lots and lots of relevant data. We then also aid in building capacity within malaria endemic countries so that they don't need to ship their sequences all the way over to the Sanger Institute in Cambridge. They can do sequencing within malaria endemic countries. So if we can aid in building this capacity for sequencing within malaria endemic countries, we can remove these shipping costs, we can remove the shipping time, and we can actually get closer to delivering real-time surveillance information. And then thinking about geographic coverage, this kind of ties in again with building that sequencing capacity, making sure that there are support networks within malaria endemic countries, and then additionally monitoring these false negatives that are occurring, and then potentially, you know, moving to different diagnostic methods if the false negatives keep occurring. So we've touched on the difficulties of malaria genomic surveillance. We've touched on how important genomic surveillance is for fighting and tackling disease. So now we're going to look at why we use big data to help us in the genomic surveillance of specifically malaria. So here is a bit of an intimidating diagram, but I don't want to walk you through the whole life cycle of the malaria parasite. I'm just going to touch on a few bits. So we understand that a mosquito can be infected with this parasite. The mosquito then can bite a person and transmit the malaria disease. And when we say transmits malaria, what we're talking about is that little parasite that's then transmitted to the human. That little parasite then travels into somebody's liver. It will stay in the liver for a while. And then after it's spent its time in the liver, it leaves the liver and enters your blood cells in your blood. So what we can do as scientists is we can take blood from a human, and then within their blood, if they have malaria, they will have these little parasites. So we do our genomic surveillance on these little parasites in my specific team, and we look at information in that organism's genetic material. So just to highlight, you can also do genomic surveillance on the mosquito, and there's a lot of streams of work within my unit working on the mosquito, but I'll be focusing just on this parasite for the rest of the presentation. So not thinking about the picture anymore, thinking about the little parasite. So the parasites have their own DNA sequence, they have their own genetic material, and they have lots of genes. So genes, segments of DNA that code for a certain useful thing, usually a protein, in the parasite that are of interest for malaria control. So we want to access bits of the genome that we're really interested in, because that information can help us improve global health. So there are lots of genes, and we can either decide to sequence all of the parasite genome, even though I mentioned that it's pretty big compared to COVID, or we can sequence parts of that parasite genome, which has really good use cases when we're thinking about providing real-time data. But both of these sequencing techniques, either sequencing all of the genome or sequencing a segment, are both very, very helpful and very valid. But a really take-home message as well is that it's only when we have the global picture of parasite genetics that we really have the power to create resources and products that are actually helpful and effective. So I mentioned that we can either sequence all of the genome, or we can just sequence those smaller segments that we know that we're specifically interested in. But it's key to note that both of these sequencing types are valid, and they're both very helpful, and we need them both because they inform each other. So if we have the whole genome data, so that whole genome of the parasite, we can do different types of analysis that are very important for malaria control. So with the whole genome data, it means that we can do discovery analysis, so we can discover new mutations that we're interested in that we previously didn't know about. We can also have a higher resolution to look at relationships between different parasites. So mainly better resolution and more discovery focused analysis. But then using what we learn from our whole genome data, we can then use that to then create these more targeted sequencing approaches where we just sequence those segments that we're interested in. And we call this targeted amplicon sequencing. So targeted, we're just targeting those regions of the genome that we're interested in. And then this is a life cycle. So if we can use this targeted sequencing approach, we're suddenly getting lots of information in real time. This could then help inform things about whole genome data, and then whole genome data can then inform how we develop amplicon targeted sequencing. So we need both. Both are fantastic, and they both feed into each other. So what are the actual use cases when we're talking about genomic surveillance for malaria? So there are many, many use cases, and I'm just going to highlight a couple on this slide. So in terms of the parasite, tracking drug resistance mutations is a very important use case of genomic surveillance. Also, monitoring changes in the diversity of genes that are targeted by vaccines. Detecting failure of these rapid diagnostic tests. So I was saying that we often have false negatives when we're testing people for malaria, and it's because these tests are often failing. So being able to use genetics to detect if this is failing in a sample is very important. And then thinking about the mosquito, tracking insecticide resistance in mosquitoes as well. So these are just a few use cases of genomic surveillance, but I'm just going to focus for the rest of the talk on drug resistance mutations. And this is because this is one of the key things we focus on in the team I work in. And it's also just very, very important when we're trying to stop people dying from this disease. So drug resistance. This slide is a bit busy. You don't need to understand the names of the drugs. I'm just trying to illustrate a picture here. So here we're looking at drug resistance, and along this horizontal axis, we're going through time. So 1940 here, progressing all the way up until 2010. So this is the time axis. And then every single colored bar is an anti-malarial drug. So we can see here over time, we've developed several drugs that are then released, to the community, able to be used to treat malaria. But the take home from this slide are these black triangles here. So the black triangle denotes resistance in the parasite was first reported to these drugs. So you can see that for nearly every single anti-malarial drug, we develop resistance. We develop resistance, 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 resistance. You see the story. So parasites evolved to become resistant to the drugs that we use for malaria very quickly. And what's key to bring up is we have a lot of drugs on this slide, but it takes a very, very long time to develop malaria drugs, push them through pharmaceuticals and get them actually into usage in country. So we don't just have lots of drugs, we can keep adding, and resistance develops fast. So we want to monitor this using genetics. And here's a story about chloroquine. So chloroquine is an anti-malarial drug, and resistance emerged first of all in Southeast Asia. So you can see here highlighted in the purple, and then it then spread from Southeast Asia across to kind of Oceania way, over India, into Africa and throughout Africa, and then independently in South America. So we had chloroquine, and then we had resistance to chloroquine starting in Southeast Asia, known as the epicenter of drug resistance, and then spreading through the world and into Africa. So this was devastating for the use of chloroquine, because the parasites were evolving against this drug, chloroquine. And this is a very typical transmission pattern of drug resistance when we're thinking about the parasite. So now I want to talk to you about artemisinin. I know these words are funny, but stick with me. So artemisinin is currently the most common treatment for malaria across the world. So in combination with some other drugs, artemisinin is the most common treatment for malaria worldwide. And recently, resistance was found in Southeast Asia. So we just saw on the previous slide with chloroquine, we know what happens if we find resistance in Southeast Asia and let it spread across the world. It will spread across the world if we don't intervene or understand how this is being caused and tracking the spread. So the community, when this evolved in Southeast Asia, were very concerned when and if this would then spread into Africa. But what's really good about genomic surveillance is that we know for artemisinin, if there are certain mutations in a specific gene, we know that that means that parasite is partially resistant to our treatments. So it means that we can use this targeted sequencing on the parasite, looking for those mutations in this certain gene. And if we find one of those, we know that that sample is resistant to artemisinin. So we can then, if we have big data, if we have a global view, we can see the spread of artemisinin resistance from Southeast Asia across as it spreads into Africa. So targeted sequencing, monitoring this in real time, we need to know when this is coming to Africa. And the answer to that is now. So sadly, we're now seeing mutations in this gene in Africa. And it's genomic surveillance that is giving us a really defined view of how, where, and when this resistance is coming from. So the gene in question is called CALCH. And mutations in Kelch mean that that parasite is resistant to the drug, and therefore that person will remain sick and not be able to clear that parasite. So here is a schematic of the Kelch gene in the dark blue. And what we can do is we can do this targeted sequencing of that Kelch gene and look for those mutations. So we can get little targets, we can put them onto the gene, and then we can sequence the genome of that gene, and then we can look for those mutations. And then if we spot a mutation in this gene in that sample, we know that if it is a mutation that changes a protein, we know that this sample could be resistant to artemisinin. So this is a really fantastic, well, sorry, not fantastic, but this is a really great way to monitor changes in real time of the parasite evolving against the drug pressures that we're applying because we're trying to treat people with this disease. So sequencing these targeted regions of the genome is more easily done in malaria endemic countries because it's cheaper, it's faster, it produces smaller amounts of data, and it's easier to analyze. And so being able to sequence in country, being able to sequence these smaller segments of the genome that tell us things about the epidemiological things we're interested in means we're closer to this goal of real-time surveillance. And one mutation alone in one sample tells you something, but it's only when you've got the bigger picture, you've got that bigger global coverage, that we can really see a different picture of what's happening in terms of, in this instance, the spread of artemisinin resistance into Africa. So to summarize, the COVID-19 was a very important example of when scientists can come together with a shared vision to tackle a global problem. But what we forget is malaria is also a global problem, but surveillance and treatment and eradication, they're affected by many, many pressures that we didn't have in the COVID space. So we need to have this global surveillance of this changing malaria parasite in order to successfully eradicate the disease and give effective treatments. So by doing the sequencing of the genome of parasites and monitoring these changes, we get closer to understanding how people will react to certain drugs, where certain resistance has come from. But one sample alone tells us something, it doesn't tell us everything. And we need sequencing at scale to see the bigger picture and to stop this disease killing as many people as it does. So with that, I would like to wrap up. I'd like to acknowledge everybody at the Genomic Surveillance Unit that I get to work with, and our work in the parasite team is supported by many teams. I'd like to thank as well all of the authors who contributed to our Malaria Gen Partner Study. So this is a fantastic resource that we're able to work with to help the community. And then a special thanks to everyone in the malaria analysis team working on parasite genomics, past and present. So with that, thank you so much for your time and attention today, and I would love to take any questions that you might have.
Fantastic. Thank you very much, Georgia. Anyone who would like to ask some questions, I'll send them to the Q&A box. Please do type the questions in there. We'll be sharing them and asking them to Georgia in just a second. We are coming up to 5:30, so if you need to leave us at 5:30, that's completely fine, but anyone who would like to stay for some questions, we'd love to pass those over. I've got a first one. I can see a few coming in. Georgia, if people wanted to learn more about the Genomic Surveillance Unit and the work that you do in malaria, where should they go to? Where should they look for that information?
So if you head over to the Sanger Institute website, there is a team page for the Genomic Surveillance Unit. So there's a brief overview of the work that we do there. And then we're also quite big on social media. So you can follow us on LinkedIn, where we post about what we get up to. And then we also have a malaria-specific social media account for Malaria Gen, which again posts all of the fantastic work that our partners get up to and that we get up to as well.
Brilliant. We'll share those social media handles out with everyone in the email after the event. I'm going to get to some of our participant questions now, because we've had some nice ones come in. So first off, I've got one here that goes way back to your earlier mentions of your career. And that was, what was your starting point when teaching yourself how to code?
Okay. So I get asked this a lot of the time, and I promise they do not sponsor me. My first thing I did was I went on Code Academy. So Code Academy have loads of really great resources, and they have an online GUI that you interact with, and it for me, it was a really fantastic website for learning to code. So I started doing Python as my first coding language, which I'd recommend to anybody because it's known to be the most intuitive to learn programming language. So I did some basic Python and then some basic command line, and then just took some other random opportunities that ended up coming my way.
Great, thank you very much. We've got a few more specific science questions here. So I've got the first one here, which was your earlier graphic on, sorry, by the way, if anyone can hear some strange noises in the background, my dog has just arrived home, so that's what, she's making some noises in the background, sorry about that. We've had your graphic showed significant fall in deaths across all age groups after 2010 in malaria. What was the cause for this? Do you have a good answer for that one, Georgia?
I mean, so generally the cause of the deaths falling was the measures that we've been able to, deploy in country. So I mentioned that we have those insecticide treated bed nets. So they've been fantastic at lowering the death rates. And then also the other malaria antimalarials that we've been able to apply. So they were the main reasons for the falls in the death, which has been fantastic because it's been shown that these measures are effective. But this is why we need genomic surveillance, because when they fail, we need to know about it so we can change these measures that we apply on those malaria causing organisms.
Yeah, great, thank you. Is there any possible genetic approach to prevent drug resistance?
Prevent drug resistance? I'm not really sure I can effectively answer that question. I don't want to give a waffling answer on that. A lot of the work we do is, trying to monitor it rather than fix it. So I'm afraid I don't have an answer for you.
Thank you for that one, Georgia. We've got something that is perhaps in a way connected here. Do you think the ongoing vaccine administration in West Africa will provide a sustainable solution?
So this is just my personal take, remember. I mean, I think the fact that we have a vaccine rollout now is a fantastic move for, trying to eradicate this disease. But we do need to remember that, the vaccine efficacy is, comparatively low compared to other vaccinations that are out there for different diseases. And currently, we're not monitoring the changes of, certain things genetically during that rollout yet. So it's going to be interesting to now have this data and be able to look at whether things in the parasite change after having the vaccination pressure applied or not. I think it's hopeful, but I still think there's a long way to go.
So there's something really interesting just to pick up on that there, Georgia, that you're saying that the vaccine itself creates a different pressure on the virus that you'll pick up the signal of within your surveillance work, so you can track how that impacts on the malaria population. Yeah,
we definitely want to do
that. We've got a question here which looks like it might be from a fellow scientist, which is there a platform or site that shares pipelines or workflows of work done on malaria that you prefer to use at your lab?
So we in Malaria Gen build our own pipelines for processing our malaria data. So these aren't open source at the moment. But we do have papers that describe the methods that we've used. So you can definitely read the Malaria Gen data release papers, because in the methods section, they explain all of the bioinformatics that we do on that data.
And you were talking about real-time surveillance, but I would like to ask if genomic data would be useful for retrospective studies. Can you give examples of situations where it can be useful?
Yeah, so retrospective studies are very...
I think we've just lost George's internet signal there, so I will mention one example of a retrospective study that I know, and hopefully we can get her back online. I'll ask Sam perhaps just to message her to see if she can exit and rejoin.
So things that this might be helpful for is if you have historic data, then you can, if you know that suddenly you've got the information that something is now resistant, you might want to go and find out when that signal first appeared and how long it took from when you first found that signal of resistance to when that was then overwhelming all of your data and all of your parasites are resistant. So yeah, you just got you've got a wealth of information looking back in time. So very helpful.
I can also say on that one, Georgia, I think we lost your signal briefly, but I have a colleague, Petra, on the team who is focused on studying the mosquitoes rather than the Plasmodium parasite itself, as Georgia mentioned at the start. That's an alternative surveillance method, and Petra looks at historic mosquito samples. So she's the closest we've got to Jurassic Park on our site, I think, in that she's getting old mosquitoes from museums and checking on their genomes to see what that can tell us about how the populations have changed too. I think we have time for just one more question before we wrap up. So that was, are you worried about old viruses coming out of the permafrost due to global warming? And I'll just add on to that, I suppose, what can we do to keep our surveillance eyes on them?
I mean, global warming affects many things, and the extent to which it affects a lot of things in science, we're still unable to like really validate that. We'll really clarify that. But yeah, I think in terms of doing surveillance, we used to do in the pandemic just surveillance on COVID, but there's a lot of work now in Sanger to be doing surveillance on not just COVID, but other, viruses within a certain respiratory sample. So you should definitely look at the RVI, Respiratory and Virus Initiative at the Sanger Institute, because yeah, we're going to be looking more in detail about what is in these samples. So we can do genomic surveillance on more than just COVID, everything else that's circulating in that. So not too worried if we're keeping tabs on it with surveillance.
And I suppose just as an last add-on to that question, Georgia, do you see a global warming signal in the way that the malaria populations are changing and adapting at the moment? Yeah,
so I think there is a lot of people working on understanding the effects of global warming on the malaria parasite. I mean, we know that as the globe continues to warm, then the chance of us having malaria in our country in our lifetime isn't out of the question as temperatures continue to rise. So there's definitely the instance of malaria, parasites and mosquitoes being able to survive in different places in the world, but also the different pressures that puts on their life cycle. And there's a lot of other people in different institutes researching this at the moment.
Great. Well, thank you very much, Georgia. I'm sorry we weren't able to get to all of the questions there. There's some fantastic other ones in the chat. Thank you, Georgia, for your brilliant talk today. Thank you all for joining us. This is the last, as I said, of our talks for this academic year, but we will be back with more Genomic Lites in September. And so we hope to see you all again there. We'll be publishing information about them on our social media channels and on our events newsletter. So do look out for that information there. I wish you all a very nice rest of your day. And thank you very much for joining us.